class. So today we will learn on the last subchapters in chapter 4, cell division and protein synthesis chapter, in which today we will learn about the genetic code and also the translation process. So remember, last week we already studied about the DNA replication, and then the copied DNA are used as a template for the transcription process of mRNA. And remember, the mRNA transcription process produce RNA transcript. And this RNA transcript is known as the pre-mRNA, not mRNA that will be used later in translation process. So what will happen? The complete RNA transcript that have been transcribed during the transcription process in the nucleus of eukaryotic cells, the eukaryotic cell will modify the complete RNA transcript after transcriptions before release it to the cytoplasm of the eukaryotic cells. So they will modify the pre-mRNA here in specific ways before the genetic messengers in the mRNA will be dispatched to the cytoplasm. So what did they modify inside the mRNA? So each of the end of this pre-mRNA here molecule, they modify in a particular way in which at the 5' end, 5' end, the mRNA, they will receive modified nucleotide of guanine bases. Meanwhile, at the 3 and prime, they will get a polyatal, meaning to say they contain 50 to 250 adenine nucleotide, only adenine nucleotide that added to the 3N. Okay. After that, remember, in the pre-mRNA, they include exon, the red color here, and also intron part. So exon is the part that contains the genetic code that will use to translate the specific protein later. However, intron part here does not contain they do contain a genetic code. However, the genetic code is not useful for the translations of the protein later. So what will happen? They will remove all this intron part here and then join back the exon part, thus completing the mRNA transcript that is ready for translation of protein. So how does the RNA splicing remove the intron? They remove the intron by using spliceosomes. Okay, what did the spliceosome do? They will cut the intron at 2 points, and then they will rejoin back all of the exon here. Thus, this part of mRNA is ready for the translation process and this dispatch or being released from the nucleus of eukaryotic cells. So let's recap back. The DNA, the double helix or double strands of DNA, they will copy themselves first under the process of cell replication during the synthesis phase in the cell cycle, and then the DNA strand will use as a template to transcribe the pre-mRNA during the process of transcription. And then this pre-mRNA is not ready yet for the process of translation, and what will happen? They will undergo alteration in which during RNA processing, they will add the guanine nucleotide type cap here at the 5 end, and then they will add also the poly A tail, which is adenine nucleotide at the end of the 3 and at the 3 end prime here. And then this mRNA will be dispatched from the nucleus into the cytoplasm of eukaryotic cell for the process of translation. Remember, all of this only happens in eukaryotic cells, because in prokaryotic cells they do not have a nucleus, and the mRNA is produced at the cytoplasm in prokaryotic cell, and they do not produce pre-mRNA. They produce directly mRNA that ready available for the translation process. So remember, inside the mRNA consists of genetic code, okay, contain genetic code, or they carries genetic code from the DNA. So what is genetic code? Okay, here they will translate the genetic code into the protein code. So what is genetic code? Genetic code is almost universal for all organisms, meaning to say all organisms mostly they have the same genetic code. So the genetic code is the overlapping group of non-overlapping groups of 3 mRNA nucleotide. 3 mRNA nucleotide. The nucleotide here is the adenine, guanine, or adenine, guanine, cytosine, and uracil nucleotide that known as a codon. And between these 3 nucleotides, between the codon, there is no space separating the neighboring codon. I will explain to you later. So what is codon actually here? So codon is a triplet basis on the mRNA strand. Okay, there is a triplet basis here. So see this one. The first 3 bases known as the first codon. The second 3 bases is the second codon, and so on. Looks, there is no space between this neighboring codon. So there are 64 possible codon that code for 20 amino acid. Remember, amino acid is the monomer for protein. Okay, so here we have 64 set of possible codon. Okay, you can calculate by yourself, and then all of these 64 is code for only 20 amino acid. We have phenylalanine, leucine, isoleucines, and then valine here, alanine, threonine, proline, serine, tyrosine. We have histidine over here, glutamate, asparagine, lysine, aspartic acid, glutamine, and so on. Okay, so however, not all of the amino acid have equal number of codon coding in for it. So for example, the tryptophan has only one codon, which is UGG. So let's look here. Where is tryptophan? So here, this one is tryptophan. So as you can see here, tryptophan only have one codon that code for amino acid tryptophan. Meanwhile, leucine has 6 codon that code for amino acid leucine. As you can see here, UUA, UUG, CUU, CUC, CUA, and CUG are the 6 set of codon that code for amino acid leucine. Okay, and there is only one start codon, which is AUG, that corresponding to amino acid methionine. So methionine, this one, okay, AUG is the start codon that code for the methionine. Start codon meaning to say that the codon that used to start the translation process of protein. And we have stop codon here, UAG, UUA, and UGA. I always call it as UA, 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 UGA. Okay, so here we have UAA, UAG, and also UGA are the stop codon, set of stop codon that used to stop the translation process or as a signal to stop the translation process later. So next, codon have its opposite side. Okay, so known as the anticodon. However, anticodon is not located at the mRNA, but anticodon is located at the tRNA, which is transfer RNA. So anticodon is a sequence of triplet base on transport RNA, on transfer RNA, tRNA, that is complement to the codon, and later they will combine with codon on the of the mRNA during the translation process. So here, so how does the flow of the coding information or the genetic code? Okay, the flow of the genetic code known as a central dogma that proposed by the Francis Crick, in which the DNA, the coding for genetic code inside the DNA, will be transferred to the RNA during the transcription process, and the genetic code that carries by the RNA will be translated into protein for protein synthesis during the translation process. Okay, so the association between DNA, RNA, and protein at the molecular level is given in this example. Okay, this is the example here. So this is the template, DNA template, which is actually over here we have 5' to 3' for the first strand, and this is the example of the second strand of the DNA that used as a template to transcribe the mRNA. So 3' always opposite with the 5'. So we have A, CC, AAA, CCG, AGT, and 5' over here. They will transcribe mRNA, which is 5'. A will form hydrogen bond with uracil. Remember, RNA do not have thymine. Okay, they only have uracil. So adenine always pair with uracil. Cytosine always pair with guanine. Cytosine with guanine. Adenine with uracil. Cytosine with guanine. Guanine with cytosine, and so on. Okay, and then mRNA, this one is called for tryptophan. UGG is calling for amino acid tryptophan. Uracil, triplet uracil is calling for phenylalanine. GGC is calling for the glycine, and UCA is calling for serine. Okay, so these are the amino acid. They will a chain of amino acid that will later will form the protein that needed by the cell. Okay, so the string of the amino acid has a direct relationship to nucleotide bases in RNA and also DNA. So now we're gonna look on the important part, which is the translation process. So translation process is the production of protein or protein synthesis from mRNA template. Okay, so it involves tRNA that will bring the anticodon, that have the anticodon. They involve codon also, okay, in the mRNA. So anticodon, and also they need ribosome. Okay, remember they also need mRNA, okay, because codon is located in mRNA. So the ribosome will make the protein by pairing the tRNA that bring the anticodon and mRNA that have the codon. Actually, they will pair the codon and anticodon between tRNA and mRNA. Next, ribosome will catalyze the condensation reaction. Okay, Condensation reactions between, meaning to say condensation is referred to the dehydration process in which they will remove water to make a peptide bond between the end of one amino acid to next amino acid. And then the peptidyl transferase activity, okay, is carried out by the rRNA, ribosomal RNA. That's why you need to know about the tRNA, mRNA, and also rRNA. And then the protein is released either to the cytoplasm or to the endoplasmic reticulum in eukaryote. But the translation process that I will show to you later is the protein that being released to the cytoplasm. Okay. So this is the structure of tRNA. I already discussed about this last week, in which, okay, in the tRNA structure, at this part, they have the anticodon site here, in which this anticodon site that being circled by red circle over here will pair with the codon that available in mRNA transcript. Okay, and the functions of tRNA is that they will transfer the amino acid. Amino acid is attached here at the three prime end. The amino acid will attach here, and they will transfer the amino acid from the cytoplasmic pool here, okay, from the cytoplasmic of the cell into the ribosome here, okay, because translation process occur in ribosome. And then the next important thing is a ribosome. So ribosome is the location where the translation process will occur. So ribosome, they have small subunit and large subunit, in which the small subunit, they have the mRNA binding site, in which the mRNA transcript that being produced during the transcription process will attach to the mRNA binding site at the small subunit of ribosome. Meanwhile, the large subunit of ribosome, they contain the E site, P site, and A site. E site is referred to the exit site, P is referred to peptidyl tRNA binding site, or I call it as a packing site, and we have the A site, which is aminoacyl tRNA binding site, which I call it as the arrival site. So we have arrival, packing, and exit site. Okay, so the ribosome will add each of the amino acid that being brought by the tRNA. tRNA later will go to the packing site, and then they will continue the process of the translation. So translation process, they have 3 steps. Same with transcription, in which they do have initiation phase, elongation phase, and also termination phase. Initiation phase is the formations of the initiation complex, or to start the translation process, in which mRNA bind to the small subunit ribosome at the mRNA binding site, and aminoacyl tRNA with complementary anticodon to start codon will bind to mRNA AUG. Okay, so for elongation process is the continued addition of amino acid to the growing polypeptide chain. It's same with the transcriptions also, and also the DNA replication. However, elongation in translation involve addition of amino acid, not nucleotide. Okay. And last but not least is the termination. Termination stage or phase is referred to the end of translation, in which when ribosome reach the stop codon, UA, UAP, and UGA, they will release factor. The release factor will bind to the arrival site of the ribosome, and the polypeptide chain mRNA and also mRNA will detach from the ribosome. The release factor same as the terminator, in the transcription process. So let's look more detail on each step in translation. So as you can see over here, okay, remember this is the small subunit of ribosome, large subunit of ribosome, and mRNA transcript from transcription will bind at the mRNA binding site over here, okay, and then this one is referred to AUG, in which AUG will call for UAC, in which it bring the amino acid methionine amino acid, okay, to start the translation process. So mRNA bind to small ribosomal subunit at mRNA binding site, and initiator of tRNA here, the first tRNA that come, they bring with the complementary anticodon here that will pair anticodon red, blue in color. They will pair with the start codon red in color at the mRNA. Then the pairing will cause tRNA and mRNA to bind to each other, and this tRNA carries amino acid methionine, met, okay, because remember in the genetic code, here AUG is calling for methionine amino acid, okay, so AUG calling for amino acid methionine, so the one that will bring the amino acid is tRNA. Next is that the arrival of large subunit of ribosome to complete the initiation complex. Remember, before before we start the translation, tRNA is the one that will bind first before this large subunit of ribosome will arrive. Okay, and the initiator tRNA here is in the P site. Okay, P site, because parking is available, so they will directly to the P site. And now the arrival site is available for the next tRNA that will bring the next amino acid. So next is elongation. Elongation step including three small process, which are codon recognition, peptide bond formation, and translocation. So what happened during codon recognition? There is the second tRNA, or the incoming amino acid tRNA, they will bind to the codon. They bring the anticodon, in which the anticodon will bind to the codon in mRNA at the A site. Why they go to A site, not P site? Because P site is full, okay, with another tRNA parking site, so they will directly go to the A site. After they go to the A site, the peptide bond formation will occur. So what is peptide bond formation? So the ribosome will catalyze the formations of peptide bond between the new amino acid here, okay, and the carboxyl end of the growing polypeptide. This is polypeptide already. We have one amino acid, second amino acid, the third amino acid, fourth amino acid, and the fourth amino acid will bind with the fifth amino acid by peptide bond. Okay, so the amino acid from tRNA at the P site, they will transfer to the amino acid tRNA at the A site. Now, tRNA at the P site do not have a polypeptide chain here. Okay, so it's free from amino acid. After that, translocation process will occur. In translocation, the ribosome here, they will move one codon down mRNA. So mRNA transcript here will move so that these two RNA also will move from its original site. So A will move to P site, meanwhile P will move to E site due to the movement of the mRNA transcript. So the tRNA with growing polypeptide here, now at the A site, now translocate to the P site. Meanwhile, the tRNA, okay, at first at the P site that free with amino acid, will move to the E site and then release from the ribosome. And then the process will continue again and again and again until it reach termination phase. Okay, so now termination phase, the last step in translation. Okay, so when ribosomal reach a stop codon, as you can see here, remember to stop the translation we need a stop codon. Stop codon is UAA, UGA or UAG. So if here we have if here we have UAG, UAG will call for the release factor, and the release factor will bind to the A site of the ribosome because A site of ribosome is always receive the arrival for the new component. Okay, so they will accept the release factor. So what will happen? This release factor that being called by the stop codon, they will hydrolyze the bond between the tRNA and the P site over here, and also the last amino acid of the polypeptide change. So release factor cause the detachment of the polypeptide from tRNA and tRNA from the P site. And next, the two ribosomal subunit, small and large subunit, and the other component of translation such as the mRNA, the release factor, will be detached from each other and the assembly will be associated. So now we already have our protein here, a free polypeptide that being produced in the cytoplasm. So, what is the differences between transcription and translation? Transcription, they occur in nucleus. Meanwhile, this is only for eukaryotic cell, yeah? And translation, they occur in cytoplasm. Transcription produce mRNA, meanwhile translation produce polypeptide, which is protein. And transcription, they use RNA polymerase, DNA template, and also transcription factor. Meanwhile, translation, they use tRNA synthesis, ribosome, or known as ribosomal RNA, mRNA template, and also the release factor. Okay, so transcription start at the promoter region, meanwhile translation start at the AUG as a start codon. And transcription, the elongation phase, during the elongation phase, they add nucleotide that bring by the RNA polymerase. Meanwhile, translation, during the elongation phase, they add amino acid, not nucleotide, that being bring by the tRNA here. And for transcription, the termination step, they will stop at terminator sequence of DNA template. Meanwhile, For translation, the termination phase will occur, or the translation will stop at the stop codon of mRNA, which is either at UGA, UAA, or UAG. Okay, remember I told you that the protein either being synthesized at the cytoplasm or endoplasmic reticulum. So before this, we have learned about the translation process that occur in the cytoplasm, that being released in the protein that being released in the cytoplasmic reticulum, in cytoplasm, right? So now the protein will be released to the endoplasmic reticulum here. So how they release the endoplasmic reticulum, how they will release the protein into endoplasmic reticulum instead of cytoplasm. So the polypeptide here that bring by the tRNA, they will call for a signal peptide, and this signal peptide is calling for SRP. So the SRP will bind to the signal peptide, and then this SRP helps this SRP, they will bind to the SRP receptor protein, and then they will translocate and bring the polypeptide change or amino acid change at this translocation complex. Okay, and then the signal peptide will be removed and the protein will be released into the endoplasmic reticulum. Okay, and next things that you have to know is about polysome. So cells sometimes they make protein very rapidly because due to some conditions or requirement. So many ribosome here, okay, they will read the same message structure. So meaning to say we already produce the protein here, and then they will go to the next ribosome to produce the same protein, okay, again and again. Okay, it's like we go here to photostat one picture, and then the picture will photostat to the next machine, and then photostat to the next machine. And if we have 1, 2, 3, 4, 5 machine, then we will produce 5 copies of the picture that we have. Okay, and this structure is known as a polysome. Okay, I think that's all for translation. So let's look on the summary of DNA replication, transcription, and translation in eukaryotic cells. So for DNA transcription and for DNA replication, remember DNA replication, they will mostly they will produce the semi-conservative model in which later this DNA will use as a template for the transcription process here. Looks here. Okay, DNA is used as the template for transcription process of RNA, and then RNA, before they being released to the cytoplasm, the RNA will undergo RNA processing, in which we will add the polyethyl at the 3N, and then we will add also the guanine cap, the nucleotide guanine cap at the 5N, and then we will remove the intron by spliosome, and we will rejoin the exon together. Now the pre-mRNA is become mRNA and ready to be released to the cytoplasm for the process of translation. And then this mRNA, they will bind to the small subunit of the ribosome, and then this mRNA have a codon, start codon. They will call for methionine, that methionine amino acid that bring by the tRNA, and then this tRNA, they have anticodon that will pairing with the codon of the mRNA, and then the process will continue until the tRNA until the anticodon until the stop codon is reached at mRNA. Once the stop codon is reached here, so what will happen? The release factor will come and then determine all of these component in the translation process. So I hope you understand, guys. So if you have any question, please let me know, yeah? Okay, that's all. Thank you.